Hey, Aaron.
Hey, John.
No longer your birthday over there, I guess, huh?
I think you're allowed to celebrate it on over 2 days. I think that's allowed.
Well, that's always a question. Like, if you're on a different date in the globe, do you celebrate your birthday on where you were born or where you are?
I think Gittler family tradition is both dates are
Hi,
allowed.
Aaron.
Well,
Hi,
happy birthday
Qiufu.
anyway.
Thank you. Thank you.
Hmm.
Hi, Qiufu.
Okay, hi. So
Hi,
let me
Qiufu.
test. So let me test see my share screen work or not. Okay. Okay. Oh, does this work?
Yeah, it looks good.
Okay. Cool. So I was thinking we'll stop sharing. Okay.
One of my neighbors is a always goes for acupuncture, and she's not a scientist, and I'm always skeptical of it. But I told her to tune into your talk today.
Yes, skeptical. Yeah, all over the places.
She says she absolutely believes it, and I said, but
You
I,
know,
I told her to tune.
even it's skeptical about when all the medical intervention fails, people
Yeah.
desperate, say go to see
Mhm.
acupuncture or a lot of those alternative medicine people. Yeah.
Is there a way of putting like the needle in like the inconsequential place? Is that just as like a test?
Try test. And yeah, I don't know those medical people. They has way, what is the region to choose based on what are the symptom, but so a lot of debate, I can mention in my talk, how many people are skeptical. It's really has acupoint specificity and other. Probably I had no time to mention like the German clinical trial a decade ago. They didn't see great difference sham control versus real acupuncture. But the sham control, for example, the needling without penetration to skin. And for us, work on neuroscience, right? So one of the most dense neural network is the skin epidermis, and so it's hard,
Yeah. Right.
to design. So we'll see.
Yeah.
I'll try to convince you. Some evidence there is some kind of specificity.
Mhm.
Yeah.
Hi, Wei.
Hi, Aaron.
Hi.
Hi, Qiufu.
Hi, wait.
Do you want to test your slides out?
Oh, sure.
Yeah, thanks.
And, I can share my. Does that work?
It's a little bit. Yes. Yep.
Okay.
Well,
Okay, it's working. Mm-hmm.
Yeah. Do you have any videos to play or anything you want to test?
Oh yes. Can you see the video?
Yes.
Okay, good. And also the sound.
I don't hear the sound.
Okay.
so,
That's fine. I can just explain.
Your sound with video?
Oh yeah, the sound comes with the video.
You might need to.
Maybe I can.
PowerPoint.
Can hear it.
Right, it's long.
Hi. Sure.
Hi, Tony.
Hey, Wei, how you doing, man?
Nice to meet you here.
Yeah.
Oh, hey,
Small
Tony.
world.
Good.
Huh? Hey. Hi. Yeah, it was funny. I just saw the announcement and I was like,
Uh-huh.
I know him.
Yeah,
How you been doing?
good. Just keep this story ongoing. And recently, we're trying to get some data from the auditory cortex.
Oh, okay. That's cool.
Yeah, by wall field imaging and a 2-photon.
Nice. Nice.
Mhm.
I wonder if you can pull out at the end organ what the fiber type is that's carrying the information based on some Uli stuff with the classification of fiber types. If the same fibers are carrying multiple modes of stimulation, will be interesting. We have the in vivo working pretty well now.
Oh, yeah.
For the
I was wondering, you mean the cochlear imaging, right?
Yeah, the cochlear imaging. Yeah, so we have that working pretty well. So it should. Are you thinking about coming over for ARO?
Yeah, probably. It depends the pandemic situation.
Yeah, I know. Well, if you are, we can take you through it.
But definitely, I can follow that online.
Yeah.
If I can go there.
Oh yeah. No, I just meant we can show you the rig and the imaging.
Sure.
We're trying now some voltage sensors with Michael Lin. If we kind of characterize again both fiber types as well as the hair cell recruitment with intensities across frequency.
Oh, nice. So basically, only voltage dye can work, right? But not
No.
calcium dye.
Well, the calcium dyes will be slower, but probably brighter. The problem we've had with the calcium dyes mostly, actually, it's not about the calcium sensor. It's more about, so what we're finding is that the ROSA site is not a good site for adult cochlea. So it works really well in young animals, but it doesn't work in adults. So we've switched to this tiger site. And while we're getting that all going, so the tiger site seems better, but we can do viral injections for the voltage site. So right now we're doing that just as a way to get moving.
Nice.
But yeah, if you just want, I haven't, there is some analysis around the calcium sensors that gets you information about frequency, even though the sensor's slow. I haven't got my head all the way around it yet, so I'm not sure how well it will work for what we're doing in the frequency ranges that we need, even for firing rates. But but that might in the end be a better way to do it, just because it integrates a little more over time, so you get a bigger signal.
Yeah.
Qiufu,
Hello. Hi.
Yes.
So,
nice to see you.
great to see you. Yeah,
Oh.
for a while.
Uh,
Cannot make any trip to China for such a long time now.
that
We can't leave China. After this, I'm going to try to get my visa extended as well.
yeah.
Because I can't really leave. Are you neighbors, Qiufu, with Zhigang?
So it's Kansas Children's Hospital. My lab actually located in the Dana-Farber Cancer Institute. So in the cancer biology department. So I'm very lonely in some sense.
I see.
Yeah. So very few neuroscientists, but we have few neuroscientists here, like Ross Siegel and Chuck Style, work on those oligo-2, brain tumor, oligodendrocyte development all the time. A lot of those neurodevelopment people left, like David Walwich, Dima Schmucker, all in my department many years ago.
Qiufu, Minjie here.
Hi, Minjie. Nice to you.
Xiong.
Too bad I missed your talk last week.
Okay,
I have to give another talk at the same time.
Yeah, you did not miss much. Yeah.
No, you missed a lot. It was really, really exciting.
Yeah.
And it's on the YouTube if you link for a few more days if you wanted to check that out.
Okay, everyone should get started.
Yeah, good. Okay, hi everyone. Thanks again for coming for another week of Neuro Zoom. Really looking forward to Qiufu and Wei's talks. Before you get started, Wei, I'm just going to share a slide. So next week we have two awesome speakers, Hailan Hu from Zhejiang University, talking about how ketamine works, and we have Shangdong Fu, and that will be great. Again, revisiting of astrocyte to neuron conversion, and we'll have lively discussion, and we'll be here as long as it takes to talk through those data. And then keep tuning in. We have coming up more great talks from a wide range of speakers. And again, please let me or Zilong know if you'd like to present your research. We have lots of spots in January and February coming up. Okay, so with that, Zilong can start introducing the first speaker, Wei.
Okay, it's my great pleasure to introduce Wei Xiong. Is assistant professor in Tsinghua University. So Wei is one of the outstanding graduates from ION. So he worked with Professor Zhou Zhuan from 2000 to 2006. So after getting a PhD, Wei followed Dr. Zhuan to Peking University. After a couple of years, he moved to had a postdoc with Scripps with Erlich Mueller, starting work with ion channels and hearing, where ever since with pretty much interesting in all the mechanism on the interesting mechanism of hearing. So 2016, so we come back to China, start his own lab in Tsinghua. So where he quickly made several very amazing discovery about the role of piezo channels in ultrasonic hearing of mammals. So I believe today we will share his latest story about molecular neural mechanism of underlying the ultrasonic hearing. Okay, welcome, Wei. Take it away.
Thank you. Okay. First, thank Aaron and Zilong for organizing this fabulous NeuroZoom talk series, and also thank Zilong for the wonderful introduction. Actually, hearing is probably an old-fashioned topic. Probably Tony is not agree with me. And as a mechanism of the animal hearing has been documented pretty well. However, our recent data just show the ultrasonic hearing use distinct molecular and the neural mechanism from those used for regular hearing. It may be of your interest because most of our colleagues are using mice as an animal model to study vocalization-based social behaviors, while mice use ultrasound.
Wait, you just need to share your screen.
I shared.
Yeah.
I stopped it just to put another slide, but you just need to close the PowerPoint and then reshare it.
Oh, sorry.
No problem. Yeah, you had shared it first, and then I took it over for the introductory slide.
Oh, I see.
So just end the slideshow and then share again, and then start the slideshow.
And the slide, I have ended the slideshow.
Okay, now just do share.
I cannot get. sorry, this. Okay.
Is PowerPoint open?
It's not working?
Yeah, it's a little bit. Yep, there you go.
Okay, sorry. Sorry for the delay. I will try to be. Including mice, many animals possess the ultrasonic vocalization and hearing for their communication in the community and daily interaction with the external world. And this is also raises a very a lot of interesting scientific questions. For example, for a bat, how the bat brain processes the ultrasonic echolocating signals to navigate in the dark space and also catch the prey in a high speed. This is an example from Cindy Moore's lab. And also, for a mouse, they can just do social interaction with their partners. For this example, the mice, this male just play ultrasonic vocalization with his female partner, and finally, he succeeded in this mating event. However, we seem to take it for granted for a very fundamental question: how mammals hear the ultrasonic signals biologically. So we all know the hearing frequency just the range increase from the lower vertebrate to higher mammals, and this largely depends on the shape of the cochlea or the hearing organ, because for the lower vertebrate there is no cochlear structure, and the length of the hearing organ increase from fish to reptiles to birds and the mammals. And this also comes with a complexity of the hearing organ. In the turtle, the reptile, it's a general homogeneous hair cell structure, and the two mammals, it comes with an organ of Corti, a relatively highly evolved organ for mammals that possess one row of inner hair cells and 3 rows of outer hair cells. For the cochlear, the hair cells distributed along the coil at the apex. The hair cell detect the lower frequencies, and at the basal coil, the hair cell detect the higher frequencies. So this is also called the tonotopy principle. This principle actually exists in all ascending auditory brain stations, and just in the higher brain stations, there's less allocation for this tonotopy because they have other high order functions. And in the cochlear, if we unroll the cochlear out, the best membrane, the first frequency analyzer, can have an oscillation at the best frequency from the sound stimulation, and on the top of best membrane, the hair cells just have a active and local oscillation with the vibration of the best membrane. That's a general theory for the frequency selectivity. And this frequency selectivity most come from a protein called prestin, distributed along the lateral membrane in the outer hair cell. Prestin, a chloride transporter cloned by Peter Dallas in 2000, is expressed all along the lateral membrane of the outer hair cells, and it possesses at least two functions. One is the voltage sensor that can fluctuate with the voltage change, and also it comes with the conformational change part. And the later one has been confirmed by most recent cryo-EM data. So far, it comes with a consensus for this cocktail amplification. So the prestin protein endows the outer cell with the electromotility, and also there's some ciliary-based electromotility, but with a uncharacterized molecules, both to coordinate to provide the cochlear mechanics. So with a prestin knockout and the knocking mice, people have shown that without prestin, the hearing sensitivity has 60 decibel elevation for the hearing threshold, and also based on the laser interferometry, people have shown there is, for example, this is a 60 kilohertz recording, and in the prestin knocking mice, there is no this kind of tuning curve. And more interestingly, some phylogenetic analysis has shown the polymorphism of prestin protein have shown some co-evolution of electromotility with echolocating animals. And also this polymorphism seems to generate different function as shown by the nonlinear capacitance. However, a most recent study has also shown the prestin protein seems to have some corner frequency. It makes sense because our cell has capacitance and membrane generated capacitance. This will generate some low pass property, and also for a protein, even for prestin, it must have some limitation for its cycle by cycle voltage sensitivity. So based on this recording, the corner frequency seems at 10 kilohertz. And also from another morphological study, the ultrasonic hearing animal, it's not tightly related with the length of the cochlear. For example, this high frequency cochlear has a shorter length compared to the low frequency cochlear. So here comes our question, whether the prestin really sense the ultra high frequency in mammals. So firstly, we introduced a very common auditory brainstem response recording, but our previous common ABR recording just to put a recording electrode underneath the scalp. So by this configuration, our the recording sensitivity is only to up to 40 kilohertz. And we next just put the electrode to a bone screw implanted on skull, and this help us to get improved sensitivity up to 80 kilohertz, because we are dealing with the vocalization frequency in mice that range from 50 kilohertz to 100 kilohertz. So next, we introduced prestin knockout mice. This was generated by Xiong Liu in Institute of Neuroscience, Shanghai, by base editing. And we first checked the morphology and the ABR in the 2 months prestin knockout mice. You can see in the 2 months prestin knockout mice, there is a lot of hair cell loss, including inner hair cell. So it makes sense. We can see most of the elevation comes for the lower frequency, and for the higher frequency, And even for the higher frequency, it's almost the profound death. That means comes from the outer cell loss. So it makes sense when we go back to the previous report, there is a severe loss for the ultra high frequency range, and not just because the preskin loss of function, but also loss of the health cells here. So we go younger mice at the 1 month, and at this age, most of the outer health cells are preserved, except some of the very basal outer health cells. And in this age, mice, we can see the lower frequency sensitivity is still elevated as the 2 months mice. And but for the higher frequency range, it is similar to the control ball type. So and it just indicates the outer health cell probably provide the ultra high frequency instead of the preskin molecule itself. But we can not exclude, because this property, this possibility that outer health cells still lost here. So we go more younger animal at 3 weeks. At this age, the mice keep most of the health cells, including outer health cells. And we can see at the lower frequencies, the 3-week mice still have a elevated lower frequency range, but at the higher frequency, it's similar to the control. But I have to say, at 3 weeks, the ultra high frequency, even in the control mice, it's a little elevated compared to the 1 month, probably because the hearing, ultra high frequency hearing, is still in development. So we next seek for another pharmacological method to figure out this question, because salicylate has been known to insert in the pocket of preskin and keep locked expanded status of the outer cell. So by IP injection in vivo of the salicylate, the hearing response will be destroyed. So with this configuration, we check the hearing sensitivity in 1 month mice, and after salicylate injection, we can see at the lower frequency range, the hearing sensitivity is elevated. However, at the higher frequency range, it's generally intact. And by time lapse, we can see after injection, the higher frequency mostly keep unvariable, but for lower frequency, it's get elevated, and finally it come back at day 2, because the salicylate function is reversible. So we next introduce the acoustically associative freezing behavior, and to test whether the mice can hear different frequencies pretty well. Here we use 60 kilohertz to represent the lower frequency, and the 63 kilohertz to represent higher frequencies. So for the 1 month pressing KO, it can hear the frequencies either at 60 kilohertz or 63 kilohertz, but after 2 months, the mice cannot hear the ultra high frequency anymore. For the 1 month pressing knockout mice, if the sound was decreased to 60 decibel, the mice cannot hear. So just below the hearing threshold of the 1 month pressing, that is just another control. So with this, we further asked whether outer health cell really be responsible for this ultra high frequency sensitivity. Here we also introduce another kind of mouse line. This is a pressin driven DTR mice, also generated by Zhiyong Liu at Institute of Neuroscience, Shanghai. So we just inject, for this mice, if we just inject the DTX, the outer health cell will be largely damaged, but the inner health cell just keep unchanged. So with the ABR test, we can see it get a very similar result with the 2 months pressing knockout. Just the for the lower frequency range, it have a pretty well preserved 60 decibel elevation, but the higher frequency range, the hearing is totally lost. And here is a similar freezing behavior test. The mice at the 60 kilohertz cannot hear pretty well, cannot. So our first summary, just for the ABR test, in pressing knockout mice and pressing DTR mice, you can see pressing probably just be responsible for the lower frequency, but for higher frequencies, we still keep a puzzling situation. So our next target will be piezo2. It has been known piezo2 is a mechanosensitive channel that was cloned in 2010, and after the cryo-EM observation, we know it's a trimer with every subunit 48 transmembrane domain, and it's for the channel, it's like a 3-blade propeller, and it's super sensitive to mechanical stimulation by vertical or lateral stimulations. Interestingly, piezo2 is also expressed in outer hair cells, and specifically expressed at the apical surface, something like here. And a regular ABR test just to show the hearing sensitivity is just a change mildly. However, this test is just limited to 32 kilohertz. So here we introduce a similar PAX2 based condition knockout of piezo2, and we can see compared to the control, the lower frequency sensitivity are similar to the control, but the higher range frequency hearing is elevated in the PAX2 driven piezo2 condition knockout. We also introduced 801 driver 9. This is most specifically expressed in the hair cells, and we can get a similar result. And the freezing behavior in the 2 conditional knockout mice, they can behave well when hearing the lower frequency stimulation, but when play the 63 kilohertz cue, those 2 conditional knockout mice cannot hear the ultra high frequencies. And this loss of ultra high frequency hearing function is not due to the loss of basal hair cells, because we can see the normal hair cell body and the hair cell bundle. And also we examine the expression of piezo2 in hair cells. This is come from the piezo2 Cree drive M-cherry expression. We can see most of the hair cells express M-cherry, and with some inner hair cell also express the M-cherry. We also stain the piezo2 expression in those piezo2 GFP fusion mice, and we can see the piezo expression at the apical surface here, because piezo2 antibody really cannot work well at the adult mice. So here we just introduce RNA scope to stain the piezo2 expression, and we can see in the knockout mice the piezo2 expression is dramatically decreased. So we next wondered whether piezo2 work in outer hair cell or inner hair cells. So we have, it is well known, the outer hair cell express prestin and the inner hair cell express VGLUS3. So here we introduce another 2 mouse line, prestin Cree-ER and VGLUS3 Cree-ER. Both can pretty specifically introduce fluorescent protein expression, like here. And then we generated the 2 conditional knockout and inject tamoxifen and examine the function at the 1 month. So in the pressin knockout, pressin drive the conditional knockout, the mice cannot hear the ultrasonic Q, but can hear the lower frequency Q. For the VGUS3 piezo2 knockout, it's pretty normal for either lower frequency or higher frequency. So basically, it's outer cell provides piezo2 ultrasonic sensitivity. We further introduced another ex vivo cochlear imaging system to examine health cell specific resolution response for ultrasonic stimulation. I will be short. Just based on this system, we can see the outer health cell calcium fluorescence when delivered ultrasonic stimulation. So first, the test is just from the TMC1 knockout mice. The TMC has been proposed as the mechanotransduction channel for the outer cells. So with the TMC1 knockout, we cannot see ultrasonically evoked calcium signal in the TMC knockout mice. Also in the piezo2 knockout mice, we also cannot see the ultrasonically evoked calcium signal. So in summary with this part, based on the previous hypothesis, the cochlear mechanics is enhanced by pressin-driven somatic motility and ciliary motility. Here we just propose probably piezo2 and help bundle mechanotransduction coordinate for a stereociliary force that can drive the ultrasonic sensitivity. So in general, our mammal probably have a very limited hearing, baseline hearing. With prestin, there is an enhanced hearing sensitivity at a lower frequency range. However, with piezo2, we can get an extended spectral range. So further, we introduce.
OK， 妈妈学习早了。
Sorry. So on the top of this cochlear characterization, we also introduce some brain imaging to see what's happening in those piezo2 knockout mice. So firstly, we introduced wide field imaging of the auditory cortex on awake mice. So basically, this wide field imaging can give us a pretty good full spectrum map at all the regions, including A1, A2, and AAF. But in piezo2 conditioned knockout mice, they lost the responsiveness in the higher range. And we can just plot here, and here is the statistics. So guided by this wide field imaging, we can locate the ultra high frequency region and inject the Cal50 520 dye, and we can do the 2-photon imaging and to draw a characteristic frequency map in the imaging region. And compared to control, we can see the conditioned knockout lost the most of the ultra high sensitive selective neurons, and we can see a very specific decrease range from 32 kilohertz to 80 kilohertz. And also after the characterization of each frequency region, we can inject the CTB and retrograde trace the input neuron from synaptic neurons. So in general, it has been shown the tonotopic patterns was still preserved in the ventral medial geniculate nucleus and showed us MGV. And also at the dorsal part of MGV, there's a low frequency, and at the ventral part is high frequency. However, we can also see this when you inject the 4 kilohertz and the 3 kilohertz region at the A1. However, the 80 kilohertz, we can see the green one, it's like a C shape just around the marginal zone of the MGV. it's a pretty normal pattern here. So here is our final summary. At the cochlear, we found outer hair cells be responsible for frequency selectivity, in addition to previously characterized the precedent for lower frequency. Probably piezo2 is contribute for higher frequency, and in the higher order nucleus, and especially in MGV, the dorsal part is for lower frequency, ventral part is for higher frequency, and we found that the marginal part is probably for ultra high frequencies. So summarize this work, I would like to thank our students. So Li Jie is leading the preceding work and the piezo2 work, and together with Chen Song, Liu Shuang, and Hu Qun, and Zikai Zhao is for the brain imaging work. And that's all my talk today. And thank you, guys.
Great. Thanks, Wei. Just all of your time mostly. So I have a couple questions, maybe.
Yes.
I have one.
Yeah, please, David.
So do you use a typical Black 6 background, or do you use a coherent 23 fixed background in your studies? I've learned recently that the Black 6 has this CDH23 mutation, which impairs high frequency hearing some. Does that get in the way?
Yeah, basically we use most of our animal are in Black 6 background, but we cannot exclude other genetic background because we use a lot of genetic manipulation, so we cannot really exclude to make the genetic background clean. But we have several other control experiments, and we check the CBA mice, and we check the bulb C mice, and also we keep our most of the mice examined at the age of 1 month, at least for this age. And for this genetic background, we cannot see by modified ABR test, we cannot see much difference for the hearing sensitivity across these mouse lines.
Okay, thanks.
Sure, thank you.
Hey, Wei.
Yes, Tony.
I was just wondering, so outer hair cells are thought to provide amplification as well as tuning in general, at least at lower frequencies, I guess. So when you get to these frequencies where the outer hair cell, apparently Preston can't keep up, where do you think amplification is coming from? Or do you think the frequency selectivity is going to be broader because you don't have a tuning mechanism to go with it? Or do you think it's also the channel?
Yeah, that's also my thought. For this one, for the piezo2 function, so far we do not have a molecular mechanism yet, but our speculation is firstly, piezo2 cannot provide amplification as precedent does, and also this one does piezo2 does not follow cycle by cycle principle, and we speculate that it's probably, as you suggested, it's just a piezo2 use probably a more general detection. I mean, not just more rely on direct current model. And for more details, we still need to get more experiment to confirm.
Thanks. I mean, it's exciting to see, because I mean, is it going to be the channel kinetics that are working at 80 kilohertz?
Yeah, that's true.
All right. Okay, some more questions. Thanks. Thanks, Wei, for good. Oh, is one more people raising your hand, please? Nicholas, is that you?
Yeah.
Please.
Hello, Wei.
Hi, Nico.
I was wondering, so is this function of piezo 2 working without a hair bundle?
So far, we have no evidence to show that. And even for the immune staining, it's just come from the P5 tissues. So currently, we are generating a HA tagged piezo 2 knocking mice, trying to answer your question. And also, we are developing more cochlear imaging technology. We want to see how exactly the hair bundle works during the ultrasonic stimulation.
Thanks.
Sure.
Okay, thanks. Great, thanks, Wei. So Aaron, we should move to Qiufu's talk.
Sure,
Great.
thank you.
Thank you.
Yeah, thanks, Wei. And it's a great pleasure to introduce our next speaker, Dr. Qiufu Ma. Qiufu received his undergraduate degree from Fudan University, one of the top universities in Shanghai. And then he moved to California for a PhD at UCLA. And then after that, he did a postdoc with David Anderson at Caltech. And here he had an amazing run as a postdoc, and he discovered neurogenin, and he discovered it using a sort of a functional cloning approach where he used degenerate RT-PCR to look for other VHLH protein encoding genes using DRG cDNA libraries. And then he identified clones, and he showed that this expression of this clone that he called neurogenin was sufficient to induce neurogenesis upstream of neuro-D. And then he went on and knocked the gene out and the related genes in mouse. And then this launched him to becoming an assistant professor at Harvard, and he rose to the ranks. And now he's a full professor in the Dana-Farber Cancer Institute and the Harvard Neurobiology Department. As an assistant professor, he was a Pew Scholar. This is the most prestigious award an assistant professor can get. And his lab has made pioneering studies using elegant genetic approaches to map the spinal circuitries that transmit mechanical pain. And then they combine that with ablation studies, electrophysiology to work out the functional properties of these circuits. He has exciting findings about neural circuit mechanisms of acupuncture, which we're looking forward to hearing. Thank you.
Okay, I would like to thank Aaron for very kind introduction, and so also give me this opportunity to discuss our recent work regarding how the electro-acupuncture drives a specific autonomic neural pathway and to modulate inflammations. So as we all know, the acupuncture has been practiced many thousand years in China, not all over the world. You know, probably people didn't know that in the United States, we have over 30,000 licensed acupuncture doctors. Even at the Dana-Farber Cancer Institute, in the place I'm working on, we have 5 acupuncture doctors, treat, those cancer patient side effects. But still, the scientific basis regarding how acupuncture works, still today, relatively poorly defined. There's a lot of people are very skeptical about the scientific basis behind the acupuncture practice. This is not just in the United States, but also in China as well. I remember a few years ago, I attended a committee meeting sponsored by Mumingpu, organized by Mumingpu, and one of the investigators gave the acupuncture talk, function imaging. Right after his talk, a former leader of the Shanghai Institute of Biochemistry, this is the institute I come from, he made the fairly blank, comment right in front of, his face, say, anyone who believes in acupuncture is not a serious scientist. So that time, I'm right about to start to think about doing acupuncture. So I'd say I'm one of them. Oh, sorry. Somehow hit. sorry. So one of the debate when you work on the acupuncture, I think we cannot avoid to discuss sometime what's the meridian channel hypothesis or acupuncture specificity issue, which my lab's not studied the meridian channel, but still the core idea behind the acupuncture practice according to traditional Chinese medicine. So there is some kind of structures they call the meridian channel, which is supposed to be the substrate and make functional connection between among all different body parts, And this kind of functional connection allow the acupuncture stimulation in a certain body region, like acupoint, and to long distantly to modulate like body physiology. Virtually can modulate whole body, physiology. For example, stimulate acupoint in a limb region like Zhusanli, you can change the gastrointestinal motility to treat, the gastrointestinal disorders. But for many decades, of course, between China, Korea, Japan, people try to understand what's the nature behind the meridian channel. Of course, no one can really see the physical presence of such, structure. And so today, that's still fairly poorly defined. As a result of this, many people still challenge the key foundation behind this acupuncture practice. Like in the Western country, many people are still very skeptical for the presence of acupoint like specificity. So today's, I'm in my talk, my life is pure neuroanatomy. So we talk about one anatomy, it's called a somatosensory autonomic reflex. So I show some evidence, suggest that this kind of reflex pathway could providing a way for acupuncture to stimulate like a somatic tissue to modulate body physiology by driving all kind of different kind of autonomic pathway. So as I mentioned, my life had for many years to study the somatosensory system from the development to the recent functional like circuit mapping in the spinal cord. And about 5 years ago, we started to pay attention to the autonomic dimension of the pain. So this kind of aspect also fairly much understudied or very much ignored by the somatosensory neural field. So basically, when you give the noxious, painful stimulation or the stimulation threat your body's integrity, we know that all the neuroendocrine or the HPA axis or single-cell neuron will be activated, to prepare animal or human to defend the threats. And then once threats, remove, and you have a body serious, body injury, then all the autonomic nervous system, the parasympathetic nervous system will be activated to reduce your activity to promote the healing. So about 5 years ago, we are really started, when we started acupuncture study, we try to map, this, what's the organization rule for acupuncture to stimulate the somatic tissue, to activate somatosensory neuron, then to drive a specific kind of autonomic nervous system, and the contact for the modulation of inflammations. So we started work 5 years ago in collaboration with Professor Yanqing Wang's lab at the Fudan and Xiang Hongjing at the Beijing Institute of Acupuncture and Mark Sebastian. And in last year and recently, we published our first 2 acupuncture papers. And this 2 paper really started to suggest some kind of like anatomic organization for acupuncture to drive a specific autonomic pathway. So today, I'm going to show you the acupuncture effects really can be impacted by many different factors, like the body region, which could reflect some kind of acupuncture selectivity or specificity, and how deep you need it to do the stimulation and what's intensity. And acupuncture effect can be strongly influenced by the disease states. And this aspect I will not talk about it today. Many talk about this body region specificity. So the body region specificity is recognized for many years already. So in 1970s, 80s, Akio Sado worked in a Rob Schmidt lab in Germany. And then later on by the Prophet Bing Zhu and Xiang Hongjing at the Institute of Acupuncture and Mark Sebastian. Both this group of people studied showing that there is really a quite dramatic body organization. Like Acucide used pinch stimulation, while Professor Bing Zhu used electro-acupuncture to draw the same conclusion. And what here is showing that they studied the gastric motility modulation by painful pinch stimulation. Dark means inhibitions, open circle for promotion. And you can see the body organization when you stimulate the limb region, you can promote gastric motility by later on they've shown that that activity requires vagal reflex, gastric vagal reflex. When you stimulate abdominal region, thoracic region, both back and abdominal region, you can inhibit gastric motility. And this requires the activation of sympathetic reflex. If you cut sympathetic nerves, you lose this effect. If you cut vagal reflex, you lose the limb region effect. So this is Professor Bing Zhu introduced me, the paradigm 5 years ago, which I thought that's a great, paradigm. Same input, produce opposite outcome, driving different autonomic reflex. So we want to understand what's anatomic basis. So in the paper we published last year in Neuron is which this is a pathway I will focus on today. It's called the vagal adrenal anti-inflammatory axis. And this axis also shows the body region specificity. And this pathway was initially discovered by Luis Ula, published in Nature Medicine in 2014. So what they found to study acupuncture modulation of the inflammation or systemic inflammation induced by bacterial endotoxin, like LPS. So what they found that when you stimulate acupuncture at a hinding region, acupuncture later on will discuss a little more detail called ST36. And they will act as a sensory neuron, transmit sensory information to the hindbrain. We're going to activate those dorsal motor nuclei of the vagus, where the vagal efferent neuron are located. And exit from the hindbrain, leading to the activation of the chromatin cell in the adrenal gland, and release a catecholamine, in particular the dopamine, which can powerfully suppress the systemic inflammation induced by the lipopolysaccharides, the LPS, can greatly promote animal survival from like a 20% to 70% or 60 to 80%. In this paper we published in Neurons, where we find that this pathway can be already activated by low electrical intensity, and it really showing the body region specificity. For example, you can activate from the hinding region, but not from the abdominal region. So mean that when you give a 0.5 milliampere stimulation, you cannot activate vagal efferent and cannot suppress inflammation. So in a more recent paper, we just published recently, we addressing the one question: so why, what's the anatomic basis for this regional specificity? Why the vagal adrenal axis can only be evoked from the limb region, but not from the abdominal regions. So this, I want to show you that because this is a basic kind of like a rostral-caudal pattern, neuroanatomy question. For us, from the developmental background, we all know that the nervous system development is a pattern around the dorsal ventral and it's a rostral-caudal and a temporal axis. And the rostral-caudal is really one of the major controlling mechanisms to subdivide the nervous system along the anterior-posterior axis. And for many years, for Tom Jason, for example, study for this motor colon development, like I say, sympathetic spinal, sympathetic preganglion neuron, only located in the thoracic level, but the lateral motor colon is only in the limb region, innervate the axial of the skeletal muscle within the limb. And more recently, my colleague David Ginty did as a single cell sequencing in the DRG and showing that at a different axial level, the representation of the molecular defined DRG neuron also shows a difference between the limb level versus the thoracic level. So basically with this, we make a hypothesis a few years ago. So because the vagal reflex can only be evoked from the limb region, so we think there must be some kind of sensory pathway selectively innervate the tissue unique for the hinding region. Like for example, the bones, the joints, and the large skeletal muscle, which you do not have in the abdominal region, for example. So this is our original hypothesis. And how we find that molecule, fit that hypothesis is really, again, it's a really come from our years ago developmental study. So when we work on DRG neuron development, and we shown that the somatosensory system can be divided into two major branches. One is depends on the dependency on their development, or say the development on the transcriptional factor called RONS1. Those RONS1 dependent neurons all innervate the skin epidermis or hair follicle. And this will form the defensive mechanism against all the external threat. While the RONS1 transient, or we call even RONS1 suppressed, the neuron, those are expressed nerve growth factor, receptor tract A, and peptidogen. And those neurons innervate throughout the body, which we saw those is a monitor internal integrity or driving the autonomic pain when you have a actual body injury. And so we think it's, because the abdominal or the limb region, we think those are, the bone joint, those are deep tissue. We hope identify among those the deep tissue innervate sensory neural molecular marker, we can find some marker to selectively innervate those. Hopefully can find some sensory neural selectively innervate those deep fascial tissue within the limb region. So we screen almost a half dozen molecular markers of Q-line, and eventually find one of the Q-line and fit, our hypothesis. And so this called a prokinesin 2, prokinesin receptor 2, or ProKR2 Q-line, which mark a subset tract A positive DRG neuron, not labeled as a proprioceptor or the IB4 positive neuron innervate skin epidermis. At almost 60% are myelinated, expressing neurofilament heavy chain NV200. And as shown this slide, we've shown that, say this group neuron do show the axial level representation difference. They are enriched in the forelimb and hindlimb region, but much less in the thoracic level. So representation almost like, in terms of percentage-wise, express this Q. It's like 3-4 difference between limb level versus trunk level. So we thought this could be the candidate for driving the vagal reflex. So here's actually shown anatomically. So when we label those neurons and show the innovation, and what's the one feature we find is really cool, this neuron has a really heavily innervate those fascia tissue, but in limb fascia, deep fascia tissue, but not innervate those deep abdominal fascia tissue like a peritoneal membrane. And this neuron also innervates all kinds of joint ligament and all the fascia wraps the muscle close to the bone. So and it shows the axial level difference. So that's why this could be the candidate for driving the vagal reflex. So we use the intersectional genetic strategy to selectively ablate, those ProKR2 Q-labeled neuron, and without, affecting ProKR2 Q positive in any other part of the nervous system, also peripheral non-neural tissue. Then we study what's acupuncture effect. First of all, we shown that this group neuron indeed are required to drive the vagal reflex from the hindlimb region. So this is acupuncture we call it Zhushani or ST36. We choose in a human. In analogous region, which is a through like right below the knee cap, and when needle through the anterior part of the muscle called anterior tibialis muscle, 3 millimeter into the muscle with a needle tip fairly close to a nerve we call it deep pronar nerve, and this nerve innervates a deep fascia tissue around the bone periosteum or the muscle surrounding those the bone. And this stimulation can enough to activate the vagal efferent neuron located in the hindbrain, indicated by C-force induction. And this induction is a complete abolish in the ProKR2 neuron ablated mice. And this stimulation can causing the catecholamine release from adrenal gland. We know this release depends on a group of chromatin cell expressing neuropeptide Y, as published in the Neuron paper last year. And this catecholamine release by acupuncture stimulation is completely lost when we remove those proKR2 neurons. And then this anti-inflammatory effect, like induced by bacterial endotoxin LPS, and all kind of cytokine we tested, like interleukin-1 beta, IL-6, TNF-alpha, they're all reduced by acupuncture, and this anti-inflammatory effect is completely abolished in mice with ablation of this group neuron. And I told you this group neuron also enriched in the following region, and when we did it, we found analogous acupoint, like we call it Xiong, which is when the needle tip close to the radial nerve, which innervates a deep fascia like a bone periosteum. And you can also produce anti-inflammatory effect in the proKR2 neuron dependent manner. So we also did a gain of function study to introduce a channel rhodopsin into this neuron. And then when you give the optical stimulation through the hind limb ST30 region, when the shines line under very close to the nerve, the deep prominent nerve, and you can mimic acupuncture effect, can activate vagal efferent, causing the catecholamine release, and also produce a strong and like an anti-inflammatory effect. And by this in a vagal nerve dependent manner, and it can really greatly promote animal survival from 20% to 70%. And this, so mean that, so what we found that this group neuron are necessary for driving the vagal adrenal anti-inflammatory axis. And I didn't, in fact, the prokinase is labeled a mixed population neuron. So like, for example, here I highlight two group neurons. One group is actually express BMP receptor 1B. Those neurons innervate only to the hair follicle, and there's no difference between the abdominal region and the limb region. While those are NF200 too high, so heavily myelinated or high myelinated, this is a peptidogen neuron, which has a deep innervation, there's a deep fascia limb. And we think this group neuron could be driving the vagal reflex. So we did a serious experiment to test that hypothesis. So when so again, within this acupoint region with the deep prominent nerve, like and common prominent just makes the branching to make the collateral branch and a deep branch, which innervates the deep, the fascia tissue. So when we cut this deep branch, within the prominent nerve, which will eliminate the innervation to the bone periosteum, but preserves the cutaneous innervation, and the acupuncture effect, anti-inflammatory, will be eliminated. If you cut the cutaneous nerve, which you eliminate skin innervation and preserve the bone periosteum innervation, and if you skin, you pinch your skin, and the animal has a complete loss of behavioral response, meaning that's a loss of cutaneous pain pathway, and you still preserve anti-inflammatory effect. So mean that there's a deep prominent nerve is more important for for driving the vagal adrenal axis. We continue to do another control experiment. So in the posterior end of the hind leg, we know the sural nerve is located right in the midline. And this sural nerve is predominantly innervates the skin epidermis. So when we put the electrical needle flanking the sural nerve, has no effect, with low electrical intensity, which you can reduce inflammation if you insert here in the Zhushani, but if you stimulate here, the cutaneous nerve, it has no anti-inflammatory effect can be produced. So this data suggesting that we identify a group sense neuron, only labeled by proKR2 pre. Most of them are myelinated, preferentially innervate deep fascia limb, but not innervate skin epidermis or skin or the abdominal fascia tissue. And this pathway are both necessary and sufficient to drive the vagal adrenal pathway, can greatly powerfully reduce the systemic inflammation induced by bacterial endotoxin. And this data, anatomists are reviewing some kind of people, we call it like a body region specificity or acupuncture selectivity in this case. Like it's So like the acupoint stimulation when needle close to the nerve send a deep, a rich innervate to the deep fascia are effective. For those regions has no such kind of nerve, all region only stimulates a cochlear nerve has no effect to drive this pathway. And this selective entirely can be explained by the distribution of those PROK2 nerve fiber. And continue, I can show you a few other acupuncture features, like when this acupuncture selectivity, this even has partially explained the acupuncture specificity. And it's showing that, like, for example, this is in a high posterior region, like a gastrocnemius muscle. Traditionally, actually, always use it as a non-acupoint control, which do not contain traditional acupoint. And strikingly, if you look the nerve distribution, all one nerves, the PROK2 really innervates those gastrocnemius muscle. Compared to like a deep, like a muscle from anterior tibulus nearby to the bone, the density is almost tenfold difference. And if you stimulate, now you put the same needle 3 millimeters into the gastrocnemius muscle, has no anti-inflammatory effect. And also we found that the needling, how deep stimulation is really critical. I can show you another study. Within the ST acupoint, which effective to drive this anti-inflammatory pathway, but it's really depends how deep you insert your needle. So anterior tibial muscle, anatomically segregates called an axial inner compartment for the peripheral superficial compartment, which contains very different kind of like a muscle spindle spectrum. And the over neuron almost tenfold difference innovation in the deep for the superficial compartment. If you insert the needle to the superficial, pull the needle out, like at this angle, insertion 3 millimeters into right underneath the skin, has no anti-inflammatory effect. So I was here's getting late. I want another fact really influence the acupoint effect is which published last year in the Neuron. It's a really intensity dependent, it's really crucial. Of course, we all know that sensory neuron stimulation with different intensity, we're going to activate different spectrum of the sensory afferent. And we found that for the vagal adrenal pathway, low intensity is enough. While for the another pathway called splenic sympathetic pathway, you need a high intensity stimulation. For this pathway, it has no acupuncture selectivity, both like an abdominal region or limb region can drive this sympathetic reflex. So what I'm told you today is basically is this or the current ongoing study by mapping the somatosensory autonomic reflex, we do show that there is some kind of anatomic organization for acupuncture to drive some specific autonomic pathway. And acupuncture space always is hotly debated. For us working on neuroanatomy, of course, any body region stimulation going to activate some sensory afferent and produce sensation. So at the end, what's the acupuncture specific mean is really as an operational definition, which has called really depend what you are measuring. For example, there's a vagal adrenal anti-inflammatory axis. It really has some kind of acupuncture selectivity. How deep, if S36 or ST25, if you high intensity low, you're going to or the stimulation intensity, it's really like if you go high intensity, any painful stimulation going to drive sympathetic reflex will not have acupuncture selectivity. And how deep, reflects like deep fascial innervation or cutaneous tissue, which we know has a very distinct sensory sensory afferent spectrum, can also drive different kind of autonomic pathway. So I will end in this talk, maybe just mention, this somatosensory autonomic is a fairly complex system. And so autonomic is virtually module every aspect of our body physiology. I said, this is a fairly complex crosstalk. And I think that we have a long way to go, to understand the full, to understand how the anatomic organization in terms of stimulate different somatosensory neurons driving different autonomic to control the different body physiology. And I think for my life to get into the acupuncture, we can discuss, if I have time, I can discuss, but it's really thinking about it, to think about a disease as it has a very complex nerve immune target tissue interaction. To develop a new therapeutic idea is have to think about it, there's a system physiology to really investigate there's a complex interaction among the nerve, immune cell, and the target tissue. And a single molecule based, strategy for the pain field so far failed. Just like a sepsis or systemic inflammation, you against single cytokine or clinical trial fail. So this is probably represent a new direction for biomedical research. So I take a little bit long time for my talk. So this is a collaboration really I mentioned from Professor Wang and Professor Jing. And Professor Bing to really introduce me those acupuncture difference in terms of, autonomic reflex. So work really done by 3 visiting scholars coming from China, Shanbing, Yang Shuai, Qiufu, and with other people in the lab, to carry out this first line acupuncture research in my lab. So I had a lot of people to help and support. I want to stop here. Thank you very much.
Thank you very much, Qiufu. I don't know what moxie bustion is, but I think it's burning some herbs or things. Okay, we have, so thanks again for the really exciting talk and agreeing to participate. We have time for questions. Khan?
Qiufu, congratulations. That is really exciting. So this reminded me during medical school, we had this video of a puncture. So it's basically needles, electrostimulation on the ears for open skull surgery. It was incredibly sort of impressive, and that stuck with me for years. Acupuncture, 4 needles in the ear, and then that, the patient he was conscious throughout the open skull operation. So, but the ear, are these kind of neurons innervate the ears? And this doesn't seem to be so deep, and this is also not on the limbs.
Yeah, we have not. Yeah, actually, so because the ear is kind of interesting because people argue there's a direct pathway from the sensory afferent to the NTS. There's one of the few regions that we call almost somatic vagal reflex, even you don't need it through the spinal cord. But actually it's an interesting area. We never checked this innovation in the ear region. It's interesting. And the concept that you mentioned, those are amazing, those are acupuncture, anesthesia study, which really part of the open up was a Western, when the Nixon visiting, China in 1972, there's a New York Times report. It's this analgesic effect by acupuncture really open up acupuncture study in a Western country. But how exactly, for example, I think very few people really do not directly do the acupuncture anesthesia only, but there is a widely practice still in Shanghai, to combine anesthesia with acupuncture so that it can reduce the dosage, anesthetic compound, can greatly promote patient recovery. But at the underlying, again, the mechanism is poorly defined. Worthwhile for study.
Thank you.
Julie?
Hi, Qiufu. That was a fascinating talk. Loved it. It's so interesting. So my question is very ignorant because I don't know very much, but I'm also excited because I'm going for acupuncture on Thursday for the first time. So now I will know what to tell them. But the question that I have is, your endpoints are very, very systemic and general. Is that a common thing that the release of dopamine then lowers TNF alpha and then everything is great, or and this isn't just from my personal thing, but seriously, how do you think of that working? It's really interesting.
Yeah, and I think, Julie, it's really nice you mentioned, because so far we use a systemic inflammation, those bacterial endotoxin, or a cycle ligation puncture, there's a model, and because this is a systemic inflammation, and we revealing this kind of some kind of selectivity. But my feeling that every organ region, like if you have a problem in the joint or arthritis, I think the organization rule we needed to re-investigate. And because even the same like a neurotransmitter, like a noradrenaline release, like you can reduce inflammation in the spleen, but sometimes can promote inflammation in the joint. So I mean that, and we had not yet we sort of labs really small lab, that we try to, but we definitely to open up those localized inflammatory model, like it's a joint arthritis, see what kind of organization rule. And we didn't touch here as a local effect, which for your treatment, a lot of people maybe more emphasize a local effect rather than systemic effect, but how the systemic dopamine can modulate the joint inflammation or something. So we don't know, but I think we needed to, really to investigate to get a better answer. Yeah.
Okay, Feng.
Oh, thank you. Professor Qiufu, I'm a student from Zhang Lab. I have a question, and is there any general sensor for acupuncture or electro-acupuncture? Cause Procare 2 positive nerve are sufficient and necessary for ST36, but not for ST25, right?
Yeah.
So I want to know, is there any general sensor for electro-acupuncture to handle it?
Not the electrical acupuncture. We know that basically there's a electric field, can activate any kind of sensory afferent, but a different sensory afferent has different threshold. Like for example, it's determined by axon diameter. Usually, it's a larger axon diameter will be threshold will be lower, and also really strongly defined by the the ion channel profile, like the potassium channel, sodium channel ratio, like will determine the electrical threshold. So basically when we say the intensity dependent, which I didn't spend too much time to discuss, but I discussed a lot in the last neuron paper. So that at least for this particular pathway, like vagal adrenal pathway, is a low electrical intensity enough. We feel maybe activate those myelinated like a delta fiber, while the C fiber activate, generally speaking, you need a high intensity. Like for example, to activate sympathetic reflex, we find that you need above 1 to 3 milliampere, for example. So it's a electrical stimulation is a kind of a way to deliver, like to activate a different spectrum of the mixture of sensory afferent. But how much would difference the manual, which then I think is another future study. So because the manual, like the first talk, like not only the PSO2 dependent, also mechanical channel, but also injury, like post-injury, like when you needle injury, we can release all those free radical or injury mediator can sensitize sensory afferent. So it's a very dynamic, what kind of sensory afferent. So we will see, for the future. Yes.
Thank you.
Okay, Lao Yushan.
Hi, dear Professor Ma. I'm Professor Jing's student. Here, I want to ask several questions. And would you think there are such neurons in human?
I'm sorry. Can you say again?
Do you think are there such Procare 2 neurons, such neurons in human?
Oh yeah. So that's a great question, because so far what we found in mice, but I think that some larger organisms, because this is the group neuron which represent a significant portion, like those myelinated a delta fiber in the bone periosteum or deep fascia. I think some overall organization rule hopefully will be evolutionarily conserved. Because the long-term persistent neuron go to skin epidermis, there's a non-peptidogic, and while the peptide go to more all over the body. This basic anatomic organization is preserved, between animal and human. But the molecular signature itself could be changed, But a functionality point of view, I think the big anatomic hierarchical organization hopefully is a concern. So we will see, and but at least, for example, in terms of gastromotility study in the Professor Bing Zhu, lab, for example, when you high intensity to from the Zhoushanli or ST36 to drive the vagal reflex to promote the gastromotility, and while abdominal region drives the sympathetic to inhibit gastromotility. That part sounds like evolution concern. Human patient can also see the similar feature. I think that we hope some basic anatomic organization, I think, should be evolutionary concern. But we need a human study to validate our funding. Yes.
Okay, thank you. And do you think it's significant to verify this kind of neurons in human? It's necessary.
Yeah, I think definitely we need someone to characterize, or maybe some even molecular marker maybe change, but this a relatively low intensity stimulation maybe can drive the same group neuron. It's a worthwhile for testing. We hope we can do some human study. Yeah, in the future.
Thank you. And I also want to ask you if there is this kind of neuron in human, and after reading the supplementary materials in that article, and it is found that the percentage of this kind of neurons in C6 to 8 and sacral 1 to 3 is higher than in the lumbar 4 to 5. And does it mean that the anti-inflammatory effect of Xiong is better than Zhusanli, as it related to the application and standardized of acupuncture in clinical? And
I think that based on our anatomic distribution, if the evolutionary concern, I think that indeed we can providing some potential guide, suggestion, I think, for the clinical study, for example, like a limb region choose against like an abdominal region, and also how deep, you need a relatively good deep needle close to the deep prana nerve in a human, and maybe use a posterior like a gastrocemic mass as a sham control site. And I think that we wish, as you guys really in China should hopefully, should start those human study. We, at this moment, we do not have the capacity, but we wish, we can do. For example, you can measure some of the patient. If I remember when the pandemic started, we really hope, we wish, actually at one point, so really wish we want to, our stimulation pattern can help to release the cytokine storm syndrome in a COVID-19 patient, or maybe for those cancer patient receives immune therapy and once a failing CAR T-like therapy. So one of the major side effect is the excessive immune response. And but I think it's almost ready for those human study based on our mouse, but we have to. Hopefully, someone can start it soon. Yeah.
Okay, thank you. Okay. And the last one, and what do you think? How different are the anti-inflammatory effects between stimulating the single acupoints like Zhusanli and multiple acupoints like stimulating Xiong and Zhusanli at the same time?
So yeah, it's possible. Like for example, like when we optimize the stimulation, like for example, Zhusanli, if it's unilateral stimulation, it's only produced like a 20 to 30% reduction in cytokine level. We need a bilateral stimulation to create a 50% reduction, so that we can see the survival rate increase from 20% to 60%. So it means that you do need some kind of dosage. So it's a possible combination like Xiong and Zhusanli will be helpful. But what's helpful, it really depends on its operational definition, because this really the artificial like a bacteria endotoxin induces systemic inflammation. But in the real world, when you have a real viral infection, bacterial infection, the inflammation had to fine tune, how much reduction of inflammation can benefit protectional beneficial, but without compromise immune system in fighting the bacterial or viral infection. So that's in that sense, I think we need a lot more preclinical and a clinical study. Yes. Okay.
Okay. Thank you, Professor.
Okay.
Oh, that's all.
Okay, Ian, do you have a question?
Yeah. Hi, Qiufu, can you hear me?
Yes.
Yeah, so I have a general question. Since the acupuncture has a lot of therapeutic effects, besides the inflammatory effects you mentioned, also antidepressant or anxiolytic effects according to clinical practice. So I want to hear your opinion about whether these different therapeutic effects shares a similar neuroanatomical basis or different neural basis.
That's great question. Certainly. So we have not yet touched the neurologic disorder yet, but I think my personal feeling and all the most tough chronic disease, one way or another, has some inflammation components, like including those neurologic disorder. You see the microglia astrocyte activation and also the infiltrations immune cell into all those different kind of the neurologic disorder. So the question is can we use the electrical stimulation or some kind of stimulation to modulate the inflammation? Remember the Li Wei Cai, those 40 hertz stimulation. When she studied the mechanistic at the end, it's also like a modulator of the microglia or immune cell activity. So I think that how the central immune cell, like a microglia, or the infiltrated immune cell can be modulated by peripheral, or I think needed to study it. I think my feeling is worthwhile. And for all kind of like chronic disease model, worthwhile to give some investigation. Yes, but I had no specific comment. My feeling is worthwhile. Yes.
Thank you. Really exciting.
Okay. Thanks everyone. Thanks Qiufu. Thanks.
Thank you.
Thanks Wei. Really exciting talks and great discussion.
Yeah,
thank you. Thank you, Zilong. Hi, thank you, Julie. Really good.